It's my pleasure now to introduce Jeff Horowitz from Ann Arbor. And Jeff is going to talk about inhibitor of apoptosis proteins regulate IPF fibroblast resistance to Fas-mediated apoptosis.
Okay. So I'll start by thanking the organizers for the opportunity to present today. And we've heard in a number of the talks so far today that fibrosis is a consequence of an aberrant wound repair process. So I thought I'd start off with the wound repair process. And what we're showing here is a normal alveolus, which is subjected to an injury of some sort, and that results in epithelial cell death and the disruption of the alveolar capillary barrier, along with some amount of vascular leak. That allows the influx of plasma proteins, including fibronectin, along with platelet and inflammatory cell influx, and the release of soluble mediators, and I've highlighted TGF-beta and endothelin, but there are a vast majority of other soluble mediators in this microenvironment, and those are released by a number of different cell types in that environment. Fibroblasts from a variety of sources are recruited into this environment where, under the influence of the soluble mediators and biomechanical factors, they undergo differentiation into myofibroblasts, which make, deposit, organize, and contract the extracellular matrix. And all of that is a normal part of wound healing. Physiologic repair requires that with precise temporal regulation, the epithelium becomes reconstituted and the basement membrane is reformed. All of that provisional and new extracellular matrix needs to be broken down and then organized into a physiologic scar, and the myofibroblasts need to undergo apoptosis. When something goes wrong somewhere in that process, the epithelium will fail to reconstitute itself. The myofibroblasts will continue to accumulate in that environment. They'll continue to make extracellular matrix, and the result of that is tissue fibrosis. So what we have, as exemplified in the fibroblastic focus of patients with IPF is an apoptosis paradox, whereby we see increased apoptosis in the epithelial cells and a lack of apoptosis in the recruited fibroblasts. And this has been shown in a number of papers. I've highlighted the one from Toby here, showing that cleaved PARP, which is a substrate of caspase-3 and an indicator of cells undergoing apoptosis, is limited to the epithelial cells in IPF biopsies, but there's no cleaved PARP present in the fibroblastic foci. So we've identified over the last several years the convergence of signaling pathways by two very commonly implicated mediators of fibrosis, TGF-beta and endothelin. These two different pro-fibrotic mediators, which signal through different mechanisms, converge on activation of focal adhesion kinase and PI3 kinase, which we've already heard about this morning. Each of these protein kinases is involved in the ability of fibroblasts to resist programmed cell death. But these kinases are ubiquitous, and they're involved in far more than the regulation of apoptosis. So what I'm going to discuss here is some of the potential mechanistic links whereby FAC and AKT signal to prevent fibroblast apoptosis. And in part, this is based on two studies from a couple of years ago, one from Steve Huang at the University of Michigan and one from Toby Marr. And the point of both of these is that fibroblasts are inherently resistant to Fas-mediated apoptosis. But when we treat fibroblasts with prostaglandin E2, which is well known to be an anti-fibrotic mediator, we can significantly enhance the apoptotic responsiveness of these cells. Now, in each of these papers, there was a figure that had to do with an inhibitor of apoptosis family member. So in our paper, we showed that PGE2 suppressed expression of Survivin, whereas in Toby's paper, he showed that PGE2 would suppress expression of X-linked inhibitor of apoptosis. So what are these IEP family proteins? There are currently 8 that have been described in mammals, and they are all characterized by the presence of this baculovirus IEP repeat at the amino terminus of the protein. In vitro, this is necessary for the ability of all family members to inhibit apoptosis. And how do they do that? Well, what I've shown here is that the apoptotic pathways, whether extrinsic or intrinsic, will converge on the activation of caspase-3. X-linked inhibitor of apoptosis, which is the only one of this family that is thought to actually function in vivo as a direct caspase inhibitor, inhibits caspase-3. Of course, with mitochondrial depolarization, as part of the apoptotic process, there's release of SMAC Diablo, which functions to antagonize XIAP. It binds to XIAP, preventing XIAP from its role in inhibiting caspase activation. There's another IAP, Survivin, which in vitro can inhibit caspase activation, but in vivo it's thought to act by other mechanisms, one of which is by binding to SMAC Diablo. In doing so, it disinhibits XIAP and allows XIAP to inhibit caspase activation. So one of the first questions that we asked is, are IAPs expressed in IPF? And the answer is yes. This is data from Toby's paper in 2010 showing that XIAP is very strongly expressed in the fibroblastic foci of IPF, and there was very little expression of XIAP within the epithelium. Toby also looked at Survivin in fibroblastic foci, again showing that it's there. It's expressed in the fibroblastic foci. Unlike XIAP, there's also very robust expression of Survivin within the overlying epithelium as well, and I'm not going to show this, but it's also strongly expressed in the bronchial epithelium. So we pulled some banked RNA from normal and IPF lung fibroblasts, and we did PCR to look at XIAP expression. And sure enough, we see the distribution here, but XIAP is significantly increased in many, but not all, of the IPF fibroblast cell lines. When we looked at Survivin, we see a very similar distribution in that it's significantly increased in about half of the IPF cell lines, but there's also half of the IPF cell lines that have no increased expression at the RNA level. Surprisingly, at least to me, there was no correlation between expression of XIAP and Survivin in these IPF fibroblasts at the RNA level. We also looked at 2 structurally related IAPs, BIRC2 and BIRC3, which are very similar to XIAP, and there was no difference in a subset of the IPF fibroblasts compared to the normal lung fibroblasts. So to look at the protein level, we cultured normal and IPF lung fibroblasts, and so this is representative data of the distribution of XIAP. On each of these plots, there's 1 normal lung fibroblast and several IPF lung fibroblasts, and they've all been normalized here by densitometry, and we see that most of the IPF fibroblasts, but not all, have significantly increased expression of XIAP at the protein level. It's a very similar picture when we look at Survivin. Here we see 2 normal lung fibroblasts and 5 different IPF lung fibroblast lines. And there's significantly increased Survivin expression at the protein level in IPF fibroblasts. Again, for those cell lines in which we had protein data on both XIAP and on Survivin, there was no correlation in their elevation. So we hypothesized that if increased IAP expression was part of the pathobiology of pulmonary fibrosis, that pro-fibrotic mediators should increase their expression. And that's the case. What we're showing here is normal lung fibroblasts. These are CCL210 adult lung fibroblasts. I can show you the same pictures from studies done in IMR90 fibroblasts. But treatment with TGF-beta or endothelin will significantly increase expression of XIAP. We had already shown that endothelin will increase expression of Survivin, and Sure enough, TGF-beta does the same thing. Next, we obtained antagonists of these two IAPs, and these two inhibitors of Survivin are structurally distinct. YM155 is a transcriptional regulator that suppresses the transcription of Survivin. Cayman 10625 blocks the ability of Survivin to bind to SMAC Diablo. And these two compounds are structurally different SMAC Diablo mimetics, so they will function to inhibit or antagonize the ability of XIAP to bind caspase 3. And as I walk you through this is a Western blot. We're looking at cleaved PARP here, and this emphasizes the point that fibroblasts are resistant to Fas-mediated apoptosis. When we treat cells with Fas in the presence of cyclohexamide, we see this cleaved PARP band, and that's indicative of apoptosis. When we treat with Fas alone, with increasing concentrations of a Survivin inhibitor, we see that there's a progressive increase in apoptosis. But if we treat with the Survivin inhibitor in the absence of a Fas activating antibody, there's no apoptosis, even with cyclohexamide. And it's a very similar story when we use antagonists of XIAP. Here we see that the combination of Fas and cyclohexamide will induce apoptosis. When we treat with the Fas antibody alone in the presence of either of the XIAP antagonists, we can induce apoptosis. But when we treat with the XIAP antagonist alone in the absence of PARP, there's no induction of apoptosis. So the next question is, does use of antagonists of IAPs sensitize IPF fibroblasts to apoptosis, or is their resistance to apoptosis a fixed feature of these cells? What I've shown here, and I've shown the dot plot so you can see the distribution of responses where each dot equals a different IPF cell line. As a positive control, these are cells treated with Fas and cyclohexamide. In the next two lanes, we see cells that are treated with either of the inhibitors of XIAP with Fas, and we see that a significant number of the fibroblast lines are sensitized to apoptosis, but neither inhibitor induces apoptosis in the absence of Fas-activating antibody. The story with Survivin is very similar, although there's a bit more of a distribution. Both inhibitors of Survivin will sensitize a number of the IPF fibroblast cell lines to apoptosis, but neither by themselves induces significant amounts of apoptosis. So to summarize, resistance to apoptosis is one mechanism that can contribute to myofibroblast accumulation in fibrotic lung disease. And we'd hypothesize that enhancing myofibroblast susceptibility to apoptosis represents a potential therapeutic strategy for fibrotic disease. We've shown that inhibitor of apoptosis family proteins are increased, at least in some IPF lung fibroblasts. They're expressed within the fibroblastic foci of IPF lung tissue. They're induced in normal lung fibroblasts by pro-fibrotic mediators, and they're suppressed in normal lung fibroblasts by anti-fibrotic mediators. So targeted IAP inhibition can sensitize IPF lung fibroblasts to apoptotic stimuli, and this warrants further investigation. So I think I'm right on time here. I'd like to thank Omorodala Ajayi, my technician who's done the vast majority of the experiments, our pulmonary fibrosis working group at University of Michigan, and especially Tom Sisson and Beth Moore for their help. Toby, who generously allowed me to use his Survivin immunohistochemistry, and of course Victor Thanikel, who got me launched in the laboratory about a decade ago. Thank you.
Thank you, Jeff. That was a wonderful talk. Question from Gisli Jenkins.
So, I mean, that was great. Clearly, if you're going to have a therapeutic for IPF, you're going to have to find something which differentially affects fibroblasts and epithelial cells. Yeah. So, I mean, certainly have you got any idea where the pathways diverge? What would be your therapeutic target?
No, I don't know where those pathways diverge. It's sort of a big black box, and that something happens in here, and they do diverge. Knowing that they do, I think that, as you pointed out, it's critical if we're going to move to a therapeutic that we need to know what's happening in the epithelium. Most these IAP inhibitors have been developed, again, going back to what Toby referred to as this concept of the parallels between cancer and IPF. So these inhibitors are being used right now in clinical trials in patients with a variety, mostly hematologic cancers, but also they've been shown to be effective as chemosensitizers. And they are not causing significant detrimental effects, but the epithelium in IPF is clearly not normal. Survivin itself has a very important role in cell proliferation, and which is probably why it's shown to be very robustly expressed in the epithelium overlying the fibroblastic foci. But what these inhibitors are going to do to epithelial cells that have already been injured, I don't know yet.
So can I follow up from that? So there are some molecular similarities between cancer and fibrosis, but one of the key divergent areas is in apoptosis, where clearly cancer and apoptosis and fibrosis and apoptosis are different. Therefore, are cancer inhibitors likely to be detrimental in patients with fibrosis rather than beneficial?
To some extent, I think it's sort of a chicken or the egg. The hypothesis that I have is that at some point this phenotype in the fibroblast becomes autoregulatory, and that you don't even need the soluble inhibitors. And that's why I think perhaps endothelin receptor antagonists haven't worked. I would be very concerned about using long-term inhibitors of TGF-beta precisely because of the cancer question. But we have a disease process in which the average mortality is worse than most cancers. So there's clearly a risk that this could have a detrimental effect. But until we have a better understanding in what's going on in the epithelium and what role these IAPs play, then that's not an answerable question. We're hoping to move this into mice, and I can say that XIAP knockout mice are viable. And they don't really have much of a baseline phenotype, and they've been used in other areas, so we're breeding them now.
Nice work. And I think what would really help it, if it's possible, and I don't know if it is possible, is to get cell lines from Masson's bodies from organizing pneumonia, and see if you can really have the opposite effect, because we know that those fibroblasts or myofibroblasts do undergo apoptosis. So that would help, I think, your entire story. And just to mention two other things, one of the first slides you showed of epithelial cells were actually small airway epithelial cells. They even had cilia on them. So it's just not the epithelial cells, the type 2 cell or type 1 cell, I should say, but it's also the small airway cells. And remember that, because that's one day going to be important, I think. And I think that's all I want to say.
I agree.
In the introduction, you mentioned there's, during IPF, the lungs damage and some of the fibroblasts recruit from outside, like bone marrow or whatever. So in your experiment, do you compare the fibroblast isolate from the normal lung or even younger lung versus IPF kind of or brio damage of the lung? Because they probably have a different population of fibroblasts.
Right. All of the IPF fibroblasts that we've used are from biopsy specimens taken at the time of either lung transplant or surgical lung biopsy, and they're grown as explants. It's the reason that I acknowledge that these fibroblasts come from a number of potential sources is once they're in the fibroblastic foci, I don't know where they come from. And I think that they probably represent a plurality of fibroblasts derived from a number of different sources, and I don't think that I or anyone else really knows whether fibroblasts derived from different origins have different functions within that microenvironment. I think there's a lot of work to be done in that, but everything that I've used are just explants from lung biopsies.
So do you think let's say, lung endogenous fibroblast, it's more susceptible to or resistant to apoptosis or whatever?
I don't think those studies have been done.
Okay.
My personal feeling is that once they're in that microenvironment and there are myofibroblasts, that they behave similarly. But again, we could look at fibrocyte-derived, we can look at pericyte-derived, we can look at EMT-derived fibroblasts versus resident fibroblasts, and I don't know that we'd find something different. And truthfully, as we move to a therapeutic, if they're already there, then what we're growing in the dish is at least representative of what's in the patient at the time that biopsy is done. But as to stopping recruitment of new cells and differentially altering their susceptibility, I don't know.
Thank you.